Swayam Prabha. Digital India. Educated India.
I'm Swati Tiwari. I'm a faculty at the School of Biotechnology, Jawaharlal Nehru University, New Delhi. And this project was supported by DTN Swayam Prabha, MHRD, New Delhi.
Okay, so now in the previous chapters, you have already learned about the cloning vectors, how to clone a gene, how to make DNA libraries, how to do PCR, and how to do sequencing. So now we come to the other part. So far we have not talked about proteins, we have only talked about DNA and RNA related stuff in recombinant DNA technology. So now we come to the other half of the recombinant DNA technology, where we'll be talking about recombinant protein expression. So recombinant protein expression utilizes different expression systems depending on the kind of protein that it needs to be expressed. So we'll be discussing the E. coli expression system and the eukaryotic expression systems. So in this first lecture, we will only be talking about E. coli expression systems. And in the subsequent lecture, we'll be talking about eukaryotic expression systems. And then in the next one, we'll be discussing about the gene fusions and fusion takes that are very commonly used to be put on the recombinant proteins. And lastly, we will discuss about what are the various challenges that people usually encounter while producing the recombinant proteins in E. coli. We will not discuss challenges for recombinant protein production in other eukaryotic sources because that is not really a part of undergraduate syllabus. So before we talk about the E. coli expression system, the first question that might be confusing for many students is that why really do we need any expression system at all? Maybe we can just take whatever natural source the protein is present in, and we can simply purify the protein. Why go through this entire, isolating and maybe cloning the gene, then putting it into an expression system in order to produce the protein? Because naturally the protein is being produced in a cell, right? So there are certain considerations as to why people go through the recombinant protein expression route rather than isolating it directly from the natural source. Of course, one can do that, but then it depends on the purpose for which the protein is going to be used and how much protein you really need, and whether you need to modify the protein further by protein engineering or not. So the first consideration is that the protein that you are going to study, maybe you want to do biochemistry, or you want to do the structure, or you want to make it into some kind of vaccine. You may need larger quantities that is present in its natural source. So the protein may be present in very small amounts. So for example, if you want to isolate a mitochondrial protein, and it is a mouse mitochondrial protein, for example, then you will have to isolate a lot of mitochondria from the mice, and so you may have to sacrifice many mice. And even then, this particular protein is a fraction of the total mitochondrial protein. So how much protein can you really make? So that is the first limitation of isolating the protein from the natural source. Secondly, if it is a cell line that you are working with, say for example, you have a mammalian cell line and it has a cell cycle protein and you want to study the biochemistry or structure of this particular protein, but the protein is present in small amounts, so you say no problem, I will just scale it up, I will grow a large amount of culture and then I will have enough protein. But that scaling up is a difficult process. Not only it is very expensive, but as you scale up, there are limitations as to how the culture will grow, how the oxygen will be supplied, and all those number of difficulties. So, excuse me. So to get enough raw material, the scaling up might be very difficult for the protein production. Thirdly, even before the recombinant DNA technology became so popular and commonplace, people were studying proteins. They were doing biochemistry, they were doing structures and everything, and often they were achieving these things by, very classical protein isolation techniques. But you have to remember that whereas methods that you apply to DNA and RNA are more or less similar, and DNA and RNA, they don't, after all, any piece of DNA is just DNA biochemically, and it does not differ so much except in its length and its sequence. So you can do these very common procedures for DNA and RNA. But proteins are a different beast altogether. Different proteins have different biochemical properties. And so often, one method that you apply for purification of one protein may not be applicable to the other proteins. So you may have to standardize lengthy and difficult protein purification workflows in order to optimize it for the protein of interest every time. Fourth, if the protein is needed for large-scale applications like for commercial applications, industrial applications, and medicinal use, then it may not be possible to isolate enough protein from the natural source. Right? Combined with these two concerns, of course, it may be difficult to have it in enough quantities. So you need to produce it in some organism which works like a factory for you, so that you can make large amount of that protein and purify it using some simpler methods of purification. And lastly, in case the protein needs to be altered in some way by protein engineering, so you may want to either change the specificity of binding, increase the binding, decrease the binding, or delete certain portions of the protein, and so on and so forth. Then for that you cannot really do it directly in the natural host at a large scale. So you need to first clone this gene which codes for this protein, and then you can do the protein engineering on that cDNA clone and then express it and use it for whatever purpose you desire it to. So considering all this, now I hope you understand why recombinant protein expression systems are needed to produce any heterologous protein in the expression system. So we can use a bacteria to express either yeast protein or human protein or xenobus protein and so on. So let's first talk about the E. coli expression system, which is one of the most widely used expression system to date. And the reason E. coli is so popular as a host organism for protein production is because it has a number of advantages. So let's see what are those advantages. First of all, E. coli has a very fast growth kinetics. So it grows very fast. Its doubling time is roughly 20-25 minutes. And therefore, you don't have to really wait for a long time in order to achieve the desired density of the culture for the production of the protein. Secondly, if you really have to scale up, then you want to grow any culture at very high cell density because the more number of cells, more the protein. So in E. coli, it is relatively very easy to achieve this high cell density culture. So people commonly use this fermentation technology where many parameters can be controlled, nutrients, oxygen, etc. can be added to the cultures continuously, and culture can be withdrawn continuously. So many things can be done. So the high cell density culture is key to be able to produce large amounts of protein, and because E. coli is very easy to scale up and a lot of work has been done on that, therefore it is a preferred host organism. Thirdly, the media that we use to grow E. coli can be made from material which is very easily available, it is universally available, it is also inexpensive relatively. So it's easy to make also. So the media components are not very exotic or very expensive components. You can easily obtain them at a relatively low cost. So if you want to scale up, then nothing like E. coli, because you don't need serum, which is very expensive. So if you're growing mammalian cells, animal cells, you may need serum, or you may need serum-free media. That is also relatively much more expensive compared to the media that we use for E. coli. And lastly, if you have to use E. coli as a factory to produce the protein, then you have to first introduce the cDNA for your gene into the bacteria, and this process of introducing the exogenous DNA into the bacteria is known as transformation. So transformation of bacteria, E. coli, it is quite fast and it is very easy. Even so in many students, even when you are doing your bachelor's, you may have worked in a lab where you have done this process of transforming the E. coli and growing the culture and expressing a protein at the laboratory scale. Okay, so E. coli, of course, has many advantages for to be used as a host for recombinant protein production. And so large amounts of any recombinant protein can be synthesized in these E. coli. But the E. coli that are used for these purposes in the lab, they are not just any wild E. coli that is occurring in the environment. They are all engineered strains. Okay, so they have been genetically engineered to have certain properties, and we will come to that in this lecture later on. Also, as we have discussed in previous lectures, many different expression vectors have already been constructed that can be used in E. coli. And all these expression vectors, they have been made possible because a lot of molecular mechanism of E. coli have been very well studied. So these expression vectors, they take advantage of that understanding, of the mechanisms that control the transcription and translation processes in E. coli, and make use of those properties in order to design vectors which are very useful for protein expression. Okay, and lastly, when we are using these vectors, the expression vectors, as you know, they are different from the cloning vectors, because in cloning vectors, all you need is an origin of replication and some kind of a selection marker, and you can just clone your protein. But for producing the protein from the plasmid vector, other than the origin of replication and the selection marker, we also need a strong and a regulatable promoter, so that we can induce the protein expression whenever we desire. So this strong and a regulated promoter is very important, so that it can initiate the transcription many times per minute under whatever specific conditions that are suitable for its induction. So let's first go for a very general procedure that is very commonly used in the laboratories and the laboratory scale protein production. So the first step is, of course, to transform the E. coli with a vector which contains the gene from which the protein needs to be expressed. So of course this gene cannot have introns. This is basically a cDNA of the gene which is cloned into a vector, and then that vector is used to transform the E. coli. Then you plate it on an agar plate and you pick a single colony so that it is not a mixture of colonies, and then it is grown in a culture under certain conditions. Till the OD at 600 and optical density at 600 is used as a parameter to monitor the growth of E. coli. So the OD at 600 is between 0.4 to 0.6, because when the OD is between this range, then the E. coli culture is in the log phase of growth. And it is very important that you induce the culture in the log phase of the growth, because that is the phase of the growth when the bacteria are really robust, they are in the best of health, and therefore they can actively channel all the metabolites and resources that are needed for this burden that is being put on them. Because if they have to really overexpress a protein, then they have to use a lot of their internal resources, the amino acids, the tRNA, the ribosomes, not only for their growth but also for the production of the protein. So it is a metabolic burden on E. coli. So it is very important that the culture is induced when it is in the log phase of the growth, because that is the healthiest phase of the culture. So one adds the inducer, or if there is whatever conditions the induction has to be done, that is added, and then the growth is continued, and then however long you decide to induce the culture for, you can do that, and then you analyze the samples on STS page. So this is a very common procedure. Of course, there are many standardizations in this procedure that need to be carried out. For example, you may decide to do the induction at lower temperature or higher temperature. You may need to standardize the temperature at which the best amount of protein is produced. You may have to standardize how much inducer to add, and all those kind of things have to be standardized. But once you have run the STS page, this is what you can expect to see. So this is the uninduced sample. So cells are lysed and the proteins are run on an STS page and stained with Kumasi blue. And this lane shows the culture that was induced to produce the protein that you wanted to. So now if you compare all these other bands, you can see they are more or less same pattern that you see, similar almost amount. But then you come to see and you see this big blob here. And this is not present in your uninduced culture. So this is the overexpressed recombinant protein that you have induced to be expressed from the vector. And so in general, by comparing the uninduced and induced samples, people can very easily figure out whether the protein of interest has been induced or not, and whether it has been induced in sufficient amounts or not. Sometimes this protein is not induced in such large quantities, and one may have to confirm the induction by Western blotting. And in those circumstances, you don't want that there is any leaky expression. Leaky expression means that the uninduced cells should not be producing this protein. Otherwise, you will not be able to make out the difference between the uninduced and induced culture. So how do you then go about achieving this process, which so far appears very simple, but actually any protein production is not a very straightforward and easy task. One may have several challenges. But how do you make the different choices and what correct choices depending on the protein you want to express. So there are three very major criteria that you want to consider when planning for protein production. One is, what is the kind of replicon that you would like to have in the vector? Replicon means origin of replication. Second very important thing to consider is what is the kind of promoter that is present in the vector. And of course, you also want to see what is the kind of selection marker that is present, because based on that, you may have to decide whether you can do the selection easily or not. So let's now discuss these three things a little bit more, one by one. So first let's talk about the replicon. So replicon, as I said, is the origin of replication, and it is basically a DNA sequence, and of course it will have other cis-acting elements which will be controlling this replicon, and depending on the kind of origin of replication that is present in the vector, if it's a plasmid vector, then the copy number of the plasmid that can exist in the cell is controlled by the origin of replication. So ORI controls the copy number of the plasmid. Intuitively one can think that, okay, if I have high plasmid numbers, then I will have a high yield of the recombinant protein. But one has to remember that the amount of recombinant protein that you want to produce in these cells would also impose a metabolic burden on the cells. And if there is a lot of metabolic burden on the cells, then the bacterial growth will slow down. And also, second thing that may happen is that the plasmid will become unstable. So there is something called plasmid instability, that when the cell divides, then some of the cells will be not getting any plasmid at all. So there will be plasmid free cells. And these plasmid free cells, because they do not have the metabolic burden of producing the protein of interest that you wanted to produce, they will grow fast, and then they will take over the entire culture. So your overall protein yield will then decrease. So oftentimes one has to compromise between how much plasmid number you want to have and how much protein you want to express, and the bacterial growth. So keeping these two parameters in mind, one has to judiciously choose a proper replicon. So some of the common replicons that are present in E. coli vectors are listed here. Of course, there are many more, but these are the common ones. The original one that was present in the early PBR 322 kind of plasmid vectors was the coli-1 origin. And this coli-1 origin basically can control the plasmid numbers and keep it between 15 to 20 copies per cell. There's another derivative of coli-1 which is known as PMB1, and that is a little bit more relaxed, and you can have anywhere between 50 to 60 copies per cell. But then a mutant form of this PMB1 was also engineered, and this has a very, very high number of plasmid copies per cell, up to 500 to 700 copies per cell. So if you want to, for example, you want to do a very short induction period, and you want to just express the protein for 1 hour or 2 hours and be done with it, then this will be the choice of origin for you. And another thing that may be of interest, supposing the protein does not fold properly and it needs a lot of time. So if there is a lot of protein being produced in a very short time, bacteria may find it very difficult to fold properly. So in those cases, you don't want to have such high number of plasmids. If there are lower number of plasmids per cell, the amount of protein produced per unit time will be a little lower, and maybe the protein may fold. Another consideration is if the protein has certain toxicity, then you do not want to overexpress it so much that the entire culture dies. So you may want to have an origin which has very low copy number. So one such origin is PSC101 origin, which allows only, it's a very stringent one, and it controls the plasmid to have less than 5 copies per cell. So depending on the nature of the protein, the folding properties of the protein, whether it is toxic or not, you may have to, and of course the length of experiment for how long you want to keep producing the protein in the E. coli, you may choose the origin which is most suitable. Second thing that is extremely important is the kind of promoter that has to be used. Now, if you remember in the earlier lectures, we talked about the LACO promoter. And just to refresh your memory, you remember that the LAC operon has these 3 structural genes, LAC-Z, LAC-Y, and LAC-A. And there is operator sequence and promoter sequence upstream of this operon. So this is the operon. And then there is a repressor protein called LAC-I, which will have, of course, its own promoter and controlling sequences. So the LAC-I produces a repressor protein, and this repressor protein binds to the operator sequence in the LAC operon. And when the repressor is bound, then even if the RNA polymerase binds to the promoter, it cannot go beyond the repressor bound operator, and therefore there is no transcription. However, if the lactose is present, then this repressor, instead of binding to the operator, can be sucked up by the lactose, and so repressor will bind to the lactose instead, and therefore it is removed from this operator and the inhibition is removed, and now the RNA polymerase can transcribe the gene. So this LAC operator, LAC operon and LAC-Z especially has been very useful and in plasmid creation of plasmid vectors, but this promoter is under regulation by the mechanism called the catabolite repression. So LAC operon is repressed by this catabolite repression mechanism. So what is that? Let's see that. So catabolite repression, it is a regulatory mechanism which is globally operated in the bacterial cells. Many things are under this regulatory mechanism. And essentially what this achieves is that it will inhibit the expression of those genes which are required for the use of some secondary carbon source when a preferred carbon source is present. So, you know, in environment when the bacteria are growing, then even though they may be capable of utilizing one or the other several carbon sources, their metabolic machinery may be, it is may prefer to use a certain carbon source which is preferred because it is easier and more straightforward to use it for the metabolism. So in that condition, even though some other carbon sources might be present, the catabolite repression will inhibit those, the expression of those genes. So in the context of the LAC operon, now what we see that the preferred, I'm sorry, I don't know, this mouse, okay. So the preferable source is glucose. So, when you grow the bacteria E. coli in labs, you add glucose to the medium. Now, when the glucose is present in the medium, then this enzyme called adenylate cyclase, it is inactive. And what adenylate cyclase does, it converts ATP to cyclic AMP. So when glucose is high, because the enzyme is inactive, the ATP cannot be converted into cyclic AMP. However, once the glucose is low, then this inhibition is removed. Now your adenylate cyclase is active, and therefore this enzyme will convert ATP to cyclic AMP. This cyclic AMP then binds to a protein which is known as catabolite activated protein, or in short it is known as CAP. So cyclic AMP will bind to CAP, and it's an allosteric regulator. Cyclic AMP is the allosteric regulator of the activity of this CAP protein, and then it goes and binds to the promoter region of this lac operon, and it will repress the expression of Z, Y and A gene from the lac operon. So basically then, if you're using lac-Z and its promoter and operator sequences in a vector, then those sequences are going to be regulated by the catabolite repression mechanism. So then what will be the limitations? One will be that the expression from this lac promoter will not be fully induced until all the glucose has been utilized by your E. coli. Now, if that happens, then you know that the bacteria is already gone through its log phase of growth, because at that time the nutrient was really abundant. And therefore, if the glucose is all gone, then it is already, the culture is already reaching the stationary phase, and therefore the conditions are not really optimal for large amount of protein to be made by these cells. So to overcome this limitation, people made a mutant form of lac promoter, which was called as lac UV5. So this lac UV5 is a mutant lac promoter which has a reduced sensitivity to catabolite regulation. So that even if the glucose is present, if you add the inducer and instead of lactose, people use IPTG, as I discussed in previous lectures. So this IPTG can then induce from this UV5 promoter even if glucose is already present, because lac UV5 promoter is not subjected to catabolite regulation. Now both original lac promoter and lac UV5 promoter, they do have high level of expression even in the absence of the inducer. So both of them are actually leaky promoters. And this leakiness is because you remember the lac-I gene, right? So the lac-I is present in the chromosome, so there's a single copy of lac-I, so there is only a certain amount of lac-I protein that can be made. And if you have a large number of plasmid DNA copies, then there may not be enough lac-I to block all the plasmid sequences, operator sequences, and therefore some promoters will be firing even when there is no lactose added. Okay, so these promoters, they will show the leaky expression. So even without the inducer, you will have expression. And so if you remember the earlier slide where I showed you the uninduced and induced protein gel, then it may be difficult for you to figure out whether there is any induction or not, okay? Because you're comparing the pattern between the uninduced and induced protein sample. So another problem with these two promoters is that they are not very strong promoters. They are rather weak, and therefore if you really have to produce a large amount of protein, then they are not very useful. So what are the solutions then? The solutions, there are many different ways in which scientists have addressed these problems. One is that mutate the promoter of the lac-I gene. So this is shown as lac-I superscript Q, and if you maybe later on you will get a chance to work in a lab, and if your boss comes and gives you a vector map and says, okay, we are going to use this vector, then you know how to now read that map. So if you see lac-I Q there, then you know that it uses a mutated lac-I promoter, and From this promoter, relatively large amount of lac-I protein can be made, and so it has about 10-fold higher expression of lac-I. Therefore, the leakiness of lac promoter can be controlled. Another very useful approach is to use synthetic hybrid promoters. So there are many such hybrid promoters, and the way they are designed is that they use the strength of some promoter. So for example, the tryptophan promoter is very strong promoter. So but to induce that, you need something which is easy and to induce, for example, the lac promoter. So people have designed these hybrid promoters. So they use the advantages of the lac promoter and they use the strength of the tryptophan promoter. So they this is known as the TAC promoter. So every promoter in bacteria, they have a Pribno box sequence, so -10, -35 position from the transcriptional start site. There are certain sequences. So this TAC promoter, the hybrid promoter, it uses -35 region of the trip or tryptophan promoter. So this makes it very strong promoter, and a -10 region of the lac promoter, so it can be induced by IPTG, and it is a strong promoter. That means it can fire many rounds of transcription from this gene per unit time. Other very useful strategy is to use a two-step expression vector system, and this is usually used, used with the T7 late promoter system. So many vectors use this strategy, and so this T7 late promoter, it is derived from a bacteriophage, which is a virus that can infect bacteria. So this T7 bacteriophage promoter is used in this system. And now, of course, if you have the T7 promoter in front of the gene that you want to express, then of course you will also need T7 polymerase, because the E. coli RNA polymerase will not bind to T7 promoter. So in order to do that, people have engineered special E. coli strains, and these engineered E. coli strains, they have an integrated copy of a prophage which has the T7 RNA polymerase. So this RNA polymerase is present in the E. coli chromosome as a prophage, and therefore it can produce T7 RNA polymerase. And this T7 RNA polymerase can then bind to the T7 promoter, which is present in the vector in which you have put your gene of interest. And once the transcription is induced from the lac promoter by IPTG, it will induce the expression of T7 RNA polymerase. And that is because this prophage is under the control of the lac promoter. Okay, so in the chromosomal copy, the T7 RNA polymerase prophage copy is put under the transcriptional control of the lac promoter. So when you add the IPTG, what was happening in other vectors, it was directly inducing the expression from the plasmid, but here it is inducing the expression of T7 RNA polymerase from the bacterial chromosome, which will then bind to the T7 promoter, which is present in the plasmid, and initiate the transcription from this plasmid for the expression of the protein. So this is known as a two-step expression vector systems, and it is very widely used system. Other promoters that are used in recombinant protein production very commonly are the arabinose promoter, ARAP, which is controlled by the sugar arabinose. There are also promoters which make use of some lambda-phage promoter, PL, and also there are cold or temperature inducible promoters, and one cold inducible promoter which is commonly used is the CSPA promoter. So all you have to do is bring down the temperature of the culture in order to induce the transcription of the gene from the plasmid. So after this, the third consideration is the selection marker. Which selection marker to use? And very common is to use the antibiotic resistance markers, as we have discussed before. And ampicillin and kanamycin are much more popular resistance markers, but also many vectors will use, chloramphenicol and tetracycline also. Now the ampicillin resistance, when it is used, it is the bacteria, if they are transformed with the plasmid, the recombinant cells, the cells containing the plasmid, will develop resistance to ampicillin. And this resistance is because the bacteria will produce this enzyme, which is beta-lactamase, which is present basically on the plasmid now. And this beta-lactamase will degrade this ampicillin. So, because this beta-lactamase, it inactivates the beta-lactam ring of the ampicillin. So, if you are continuously producing the protein, then continuous production of beta-lactamase will eventually degrade all the ampicillin that is present in the media. So basically your selection pressure is gone. And because of that, plasmid free cells can increase with time in the culture, and the protein production will suffer. Compared to ampicillin, the tetracycline resistance is much more stable, because it does not depend on the degradation of tetracycline, but it is basically it is effluxed out from the E. coli cell, and therefore the cells which can throw out tetracycline out of the cell will be resistant to tetracycline pressure. So that is much more stable compared to ampicillin. But when you use antibiotics as selection marker, then if you are, laboratory scale production, you may be growing at the most, 50 ml culture, 100 ml culture, or maybe at the most a few liters of culture. But if a very large scale protein production has to be used, then there is a certain limitation of these antibiotics. One is that the cost of production goes up extraordinarily high, because the antibiotics will be expensive. And secondly, if a proper disposal technique is not employed, then one can spread this antibiotic resistance, introduce this resistance in the water bodies, and which is really a very big problem for because of the environmental concerns. So the solution to this is to use antibiotic-free selection systems. And there are many such selection systems that have been developed, but they are not still very widely used, and they are not very common, at least for the laboratory scale protein production. So in order to express the proteins, there are many different E. coli strains that have been developed, and many of these strains are commercially available from different companies. And so which strain to use depends on certain criteria. And so let's go over these strains one by one. So the first strain that was introduced was the BL21 E. coli cells. And this BL21 cells here, basically the T7 polymerase is not encoded in the bacteria. It is introduced with the lambda phage. So this BL21 can bind to lambda phage, and under the control of lambda CE6 promoter, the T7 polymerase will then be produced. And therefore, the whatever plasmid, if it has the T7 promoter, the protein production can take place. So because the T7 RNA polymerase is not encoded, it is not in the genome of the bacteria, it is not present in the vector that you have introduced in the bacteria, it is coming only when you add the lambda phage separately. This is the tightest control that one can have, and there is no leaky expression in the system. But the induction with this in this system is not very efficient. So that is the so this is the pros, this is the con. Another very popular strain is BL21 DE3. Of course, it has been derived from BL21. But here, instead of lambda CA6, which has to be introduced by externally adding the lambda phage, this D3 there is a D3 lysogen in this bacteria which contains the T7 polymerase. Also, this strain is deficient in certain proteases. So, LON and OMPT protease are the chromosomal copies are deleted, and therefore this promoter, this strain has high expression from the T7 promoter, but there is a leaky expression also, some leaky expression. But because the proteases some proteases are not present, the expressed protein may be more stable in these strains. Another popular strain is BL21D3 P lysis, and here there is another plasmid in this strain which produces T7 lysozyme. And because of that, it has reduced expression of proteins because this lysozyme will take care of that. And therefore, if you are trying to, I'm so sorry, if you're trying to express a toxic protein, then you do not want to have very high expression. And therefore, this strain is suitable for production of toxic proteins. The next strain is this BL21 codon plus RIL strain. Now this strain is used when you want to express those genes from organisms which have a codon bias. So let's first understand what is codon bias. So you know that we have this universal triplet codon for all the amino acids. And you notice that for some of the amino acids, there are multiple codons, right? So for example, arginine has 6 codons here and here. Similarly, I'm so sorry. My mouse somehow works very fast, and even a small touch makes it go here and there. Sorry about that. Similarly, for isoleucine, you can see that there are these 3 codons. For leucine, again, you see there are these 6 codons, and so on and so forth. So now, any organism can use any of these codons to code for various amino acids. But what happens is that not every codon for a particular amino acid is used at equal frequency. So some codons will be preferably used at a much higher frequency in some organisms compared to some other codons. So for example, it is quite possible that an organism uses these 2 codons much more frequently, say 50% of the time, compared to the rest of the 4 codons for arginine. Now you know that when the proteins are being synthesized, then the transfer RNAs, they have an anticodon sequence which will bind to the codon, and then they have the corresponding amino acid which is present at the 3' end of this tRNAs, and if the anticodon recognizes the codon, then this amino acid will be introduced in the protein. So any organism, if it uses say a particular codon much more preferentially compared to the other one, then that corresponding tRNA will be present in larger amounts in that particular organism. So the tRNA for every codon is also not distributed at equal frequency for all the codons. So this is known as the codon bias. So for example, if I'm using any gene from P. falciparum, P. falciparum's genome has an 80 rich sequences. So it will use those codons preferentially which have more A's and T's compared to G's and C's. So corresponding tRNAs will be present in larger quantities. And therefore, if you put an P. falciparum gene and you want to directly translate that in E. coli, it may not work because certain codons and their correspond, because they are not used very commonly in E. coli. Therefore, E. coli may not have those tRNAs in abundant quantities, and therefore the protein translation will stop. So the solution to overcome this is to use certain strains of E. coli. So for example, this BL21 codon plus RIAL. This has extra copies of certain codons, the extra copies of tRNA genes which recognize certain codons which are not used very frequently in E. coli. So if you want to express those genes in E. coli which have a certain codon bias, then you can use this strain, because then the tRNAs will, for those biased codons, will be present in this particular strain, but not any other E. coli strain. So this strain will be able to produce those proteins. Another very useful strain is this Arctic Express, and there are many such similar strains from many different companies. What it does is that it expresses some cold adapted chaperonins, and so these chaperonins, they help the protein while it is being produced to be folded, and therefore if the protein of your interest does not fold and it becomes insoluble because of aggregation, etc., then this is a very useful strain because it addresses the protein insolubility problem. There are also strains which have been developed. For example, this shuffle strain. What this strain does is that it has reduced or in pathways, the reductive pathways which are present in the bacteria, they are diminished, or they have been rendered slightly ineffective. So it allows the formation of disulfide bonds in the protein that you are producing, and therefore, therefore the protein can fold, because sometimes some proteins, they will need disulfide bond formation in order to fold properly. So these are, and there are many more E. coli strains that are out there, supplied by many different companies. So depending on the kind of protein that you want to express, the kind of problems that you may be having with the protein that you are expressing, you may choose a different strain, and many times it solves the problem of protein production. So the lastly, I had told initially that the DNA is introduced in E. coli by the process of transformation. But before we transform the E. coli cells, they have to be first made competent to take up the plasmid DNA with a high efficiency. So this is known, these such cells are known as competent cells, and there are many different methods for preparing these competent cells, but one of the most commonly used method in the labs is the calcium chloride mediated method. The mechanism is not very well understood, but treatment with cold calcium chloride renders the E. coli cells more efficient in taking up the plasmids. So this calcium chloride method is the very commonly used method. And then these competent E. coli cells are transformed with the plasmid vector. This can be done either using a heat shock method, which is a very common and very easy method, or they can also be transformed by electroporation, which is using an electrical shock to create transient pores so that the plasmid can go through the membrane. So you take, this is shown schematically here, you pick a colony of E. coli, grow it, and then you make the competent cells using cold calcium chloride solution, and then you may transform it with the plasmid. So if this plasmid contains the ampicillin resistance, then you may transform it by either heat shock or electroporation, and after that you plate them on ampicillin containing agar plate. So you will have these plate, you incubate it anywhere between 12 to 16 or even up to 18 hours, but not longer than that, because longer incubation time will produce two problems. One is that if it is ampicillin resistance you're looking for, then ampicillin gets degraded, as I told you, and therefore satellite colonies will start appearing. These are very small colonies which will start appearing, and also the colonies will start fusing together, so you may not be able to pick your clone. But if you are careful, then you may have well isolated colonies, and then you can pick the colony of and then do further downstream experiments with that. So to summarize this first lecture, then I have told you that many expression systems have been developed for recombinant protein production, and in this first lecture we have developed the E. coli, which is the preferred system for protein expression because it is easy to culture, it has very fast growth, it can achieve very high culture density, and the cost of growing this culture is very low. Then there are three very important considerations when you are using E. coli. Then the vector that you use, you have to think about the what is the kind of origin of replication this vector has, what is the kind of promoter that the vector has, and what is the selection criteria for this particular vector. And there are many replicons that have been engineered in various vectors, and these various replicons can give high to low copy numbers of plasmids. So wild type coli1, modified coli1, PMB1, mutated PMB1, these are relatively high copy numbers plasmids, and this origin PSC101 controls the number of plasmid copies to less than 5 per cell. Then coming to promoters, the modified lab promoters have been engineered which can overcome catabolite repression, but these suffer from the problem that they are leaky, and also they are not very strong promoters. So hybrid promoters which combine the strength of other promoters, for example tryptophan promoter, and the ease of the lac promoter, they have been engineered to be used in various vectors. Other than the lac promoter, there are two-step systems which have been developed, and they commonly use T7 polymerase, which is integrated into the bacterial chromosome as a prophage under the control of the lac promoter. And the T7 promoter, which is the bacteriophage promoter, it is present on the plasmid. And although it does take care of the leaky expression to some extent, it is not really totally foolproof system. There are many other promoters that have been developed, and some of the temperature sensitive promoters are very useful. The antibiotic selection is the most commonly selection method used, and ampicillin is the most commonly used antibiotic selection method, but ampicillin suffers from the limitation that it gets degraded by beta-lactamase, and therefore it can give rise to plasmid-free cells that can overtake the culture and reduce the protein yield. So one has to be careful about how long you have to grow the culture, because long time growth will give rise to plasmid-free cells. Another concern about the antibiotic selection is that the antibiotics can be released into the environment if very large scale cultures are done. So that is another limitation. And there are many different E. coli strains that have been developed which help in the protein production, because these strains, they have improved plasmid stability, or they may be deficient in certain proteases, so they have reduced proteolytic degradation. They may be able to address the codon bias, and some of them also have expression of co-chaperones in order to fold the protein of interest much more efficiently. So this is the end of the first lecture, and then in the next lecture we will be talking about the eukaryotic expression systems. Thank you.